Hello guys, welcome to my YouTube channel. So today we are going to discuss one of the most important pathways of complement activation, which is the classical pathway of complement activation. Now the central component of the classical pathway of complement activation is the C1 complex. So this figure that you see is the figure of the C1 complex. Now this C1 complex is in turn composed of three different subunits. One is the C1Q, then you have C1R, and then you have the C1S. So these three subunits altogether constitute what is known as the C1 complex. Okay. Now in the figure, this sea green color subunit that you can see here, this is the C1Q subunit, and apart from that you have two other subunits. This blue color subunit that is shown, this is C1R, and there you have C1S. Okay. Now one thing you should note here, that the subunits C1R and C1S exist as a homodimer. So that is why they are also written as C1R2S2. Okay, so that means two subunits of R and two subunits of S. And this portion of the C1Q subunit, okay, so this portion that I am highlighting here, this is the portion which actually binds to an antigen antibody complex. So here, as you can see, this dark green thing that is shown, this is the antibody, and this antibody has been bound to antigen present on microbial surface. So C1Q can only bind to antigen antibody complex and not to any isolated antibody. Alright, so this is one characteristic, and here you should note another thing. The portion of the antibody which binds to the C1Q is the FC portion. Alright, so FC portion of the antibody is that portion which binds to the C1Q subunit of the C1 complex. And the antibodies which mediate this pathway, or which play central roles in activating complement by this pathway, are IgG, and you have another antibody which is IgM. So this is very important. These two antibodies are major key players in this type of complement activation. So apart from this C1 complex, you have some other complement proteins which play central roles in this type of pathway, which are C2, C3 and C4. Now let us see the detailed steps of how these types of pathway activates complement. So the first step is when the antigen antibody complex binds to the C1Q. So here, as you can see in the figure, this antigen antibody complex has been bound to the C1Q subunit of the C1 complex. So what happens when antigen antibody complex binds to the C1Q? It becomes activated, which in turn activates the subunit C1R. So one C1R subunit then activates its partner C1R subunit. So this C1R subunit then activates one C1S subunit, and this one C1S subunit, like as before, activates the other C1S subunit. Okay, so in this way the entire C1 complex is activated. Now what is the second step? This C1S subunit, alright, which has been activated in the first step, this activates another complement protein which is C4. So what does it do? This C1S subunit cleaves C4 into two fragments, which are C4A and C4B. Alright, so on the left hand side, if you see, this same thing is shown. This C4 molecule is been broken down into two fragments, C4A, which goes away, and C4B, which comes and binds to the target membrane surface. So when C4B binds to the target membrane, it becomes capable of activating and cleaving C2 protein. Okay. So what happens? C2 protein comes and binds to C4B, and C2 protein is in turn cleaved into two fragments, C2A, C2B, which goes away, and C2A, which remains bound to C4B. Okay, so as this C4B2A, and this complex is known as C3 convertase. Okay, so what does C4b? So C4b do? C4b cleaves. Okay, so C4b, what does it do? It cleaves C2 into two fragments, C2a, which goes C2a and C2b. C2b goes away and C2a comes and binds to this C4b and forms the C3 converters. Why it is called C3 converters? We will see that in the subsequent slides. But one thing you should note here, this C4b2a complex is actually made up of two types of fragments. One is C4b and the other is one is C2a. Alright. Then what is the next step? Here C3 convertase, it is called C3 convertase because it cleaves C3. That is why it is called as C3 convertase. Okay. So here as you can see, this C4b2a, which is a C3 convertase, cleaves C3 into two fragments, C3b and C3a. C3a goes away and C3b comes and joins this complex and forms C4b2a3b complex, which is also called as C5 convertase. Why C5 convertase? Same as before, because you will see that this convertase in turn will cleave C5 complement protein. Here also, like as before, this complex is formed of three fragments. One is C4b, then you have C2a, and the other fragment is C3. Alright. Now what is the last step? The C5 convertase formed in step 3 comes and cleaves C5 protein into C5a and C5b. C5a goes away and C5b remains bound to the target membrane surface. Alright. Now what is the significance of the classical pathway? So C3b, if you recall, it is at the central point of the classical pathway. Okay. So many steps come and join at C3b and many steps diverge from C3b. So it is actually at the central focal point of the classical pathway. So formation of C3b is particularly important because it binds covalently to microbial surfaces and act as molecular tags, because these tags guide the phagocytic cells to destroy these microbes by opsonization. So once you bind C3b to any microbial surface, then phagocytic cells can recognize, oh, these are the microbes which we need to destroy, and they destroy it by opsonization. Another point that C3b can do is C3b can bind to antigen antibody complex. Okay, C3 can bind to immune complexes. Here also they can function as molecular tags and guide the phagocytic cells or the red blood cells to phagocytose or destroy these immune complexes. And because C3b is formed at the central point of the classical pathway, formation of C3b is particularly important because it will join the C4b2a complex or the C3 convertase to form what is known as the C5 convertase. Because only when C3b joins the C3 convertase, it will be form C5 convertase, which can keep C5. It's okay. So formation of C3b is particularly important, and these are basically the steps of significance of classical pathway, because C3b is particularly important and it play very key roles, not only forming the C5 convertase but also functioning as molecular tags, okay, to guide the phagocytic cells to destroy microbial surfaces or antigen complex with antibody. Okay, so these are the significances of classical pathway.